Suppose we are provided with multiple sequences and then we want to construct a phylogenetic tree amongst those sequences. What is the step or what is the procedure? A common method is based on distance matrices, which follows the approach of number one, we will apply multiple sequence alignment to those sequences. Then we will put those distances in the form of a matrix, and finally we will generate a phylogenetic tree. So moving on, if I give an example of three sequences as shown in this slide, gorilla, human and chimpanzee, which are provided with some sample data. So what we see is that between human and gorilla, there is a difference of 4 characters, and between human and chimpanzee, 2 characters, and similarly for gorilla and chimpanzee, distance of 4 characters. How we can make a phylogenetic tree after this step? We have done sequence alignment, we are done with matrix for distances, and now we want to make a phylogenetic tree. So if I want to make a manual tree, it would be something like shown in the lower part of this figure. Again, for very simple or simple sequences, it would be easy to make a tree, but for heavy sequences, it would be very time consuming. So for that purpose, we will see how from given sequences we can make phylogenetic trees. So moving on, there are two type of distances. Number one distance is called observed distance. Basically, observed distance is a distance which is shown in our sequences. For example, in our previous example, we knew that between gorilla and human there were 4 sequences, were 4 characters were different, and we say that that observed distance is our distance between characters in the sequence. And the other one is our patristic distance. Patristic distance is our distance which is present in our generated tree. So patristic distance is our generated distance in the tree, and observed distance is our distance in the sequence. And our goal should be that our observed and patristic distance should be always equal. Moving on, if I want to make this distance and if I want to make a tree from provided sequences, there is a method which is called least of squares method. So how we will say that number one, we have multiple sequences and they are aligned as given by this thing. Moving on, if I want to convert it into table, so it would be something like this. We have 4 sequences S1 to S4 on horizontally and vertically. So what we are saying that there is a distance of 2 characters between S1 and S2. There is distance of 9 characters between S1 and S3 and 8 characters between S1 and S4. Moving on, unrooted tree for this information or this table would be something like this, that we have S1, S2, S3 and S4, and then there are different parameters. For example, between S1 and S2 there is two things, nodes A plus D. Between S1 and S3 we have A plus B plus C, and similarly between S1 and S4 we have A plus B plus E. So this distance is shown by D12, D13, D14 and so on. Moving on, if we want, what is our goal now? Our goal was that we want to minimize the distance between our observed distance, which is shown in the table, or between our sequences, and the other one was distance shown on our tree, phylogenetic tree, which is shown by a small d. So for that method we have sum of least square method, in which what we do, we take combination of one capital Dij and one small dij, and then we find distance between first d1, then second d1 and so on, and then we sum up those results, and there those results are shown by q. So when I do this thing, and in the next step what we do is that we have this q which is sum of our least square distances, and then I put values of d12 minus d12, d13 minus d13, and I am taking square of this information like this. Okay, after substitution of values, so substitution of values means for d12, for example, if I write here for d12, it should be something, d12 is our 2 minus d12 small one is basically our a plus d, so it would be a minus d. And if I take square root of this, and okay, so similarly I will fill all these values, and then after substitution of values I will get this information, which would be equal to q is equal to 319 minus so on. Now how can I find values of individual elements such as a, b, c, d? So for that purpose we take help from mathematics, and we have idea of partial derivatives. So what are those partial derivatives? So for partial derivatives, we will calculate individual partial derivative for A, B, C, D and E. And this would be something like this. So what we do, we take partial derivatives and make it equivalent to 0. And finally, we will get results in the form of A values of different parameters such as A would be 1, B would be 5 and so on. And now if I redraw the tree now, phylogenetic tree, it would be something like this. So there is distance of two things between S1 and S2, as we said earlier, that we have a distance of 2. So which is shown here. So between S1 and S2 we have 2. Then between S1 and S3 we had a distance of 9, which is shown by S1, S2 and S3. It is here 9. And similarly we can find out other distances. So what is the output of this algorithm then? We had our initial sequences. We aligned them using multiple sequence alignment. Then we convert them into table, and then by using partial derivatives, we have converted them in a unrooted tree which has different weights for branch which are present between the tree. So what is our conclusion? We have construction of unrooted tree with uh, weights which are assigned to different nodes and branches, and using multiple sequence alignment techniques.